Today we'll be looking at gray matter volume loss in association with specific clinical motor signs in Huntington's disease. The research was originally conducted by Emma Koppen, Mila Jacobs, Anita Evanderberg Heismans, Jeroen van der Grond, and Raymond A.C. Roos, and it is presented to you by Austin Hall. HD is an autosomal dominant genetic disorder, meaning that only one affected copy of the gene is enough to cause the disorder. So you can see it in each generation with an equal chance of it affecting both male and females. Due to the nature of only one gene needing to have the abnormal sequence, a child of a known carrier of the gene has a 50% chance of getting the affected gene passed on from a parent. HD is also a triplet repeat disorder. The defect is located in a gene called HTT, located on chromosome 4, which contains a CAG repeat which normally is repeated anywhere from 10 to 35 times. With Huntington, this CAG repeat will continue for more than 36 times. The defect messes with DNA polymerase, as it doesn't count which CAG repeat it is on, so in the primary transcript there will be an excess of CAG repeats. This full mechanism for the defect is relatively unknown, with only clues leading to the exact cause. Whether it be a problem with the DNA polymerase or a miscoding of the terminator sequence is unknown, with the added struggle that as the gene is passed, it could potentially develop more and more CAG repeats with each generation, leading to earlier onset of HD. So the parent may have only 38 repeats, but a child could develop 40. This is referred to as anticipation. With each generation that passes, earlier symptoms will start to show. The problem also arises in that CAG codes for glutamine, so people with the affected gene will have 36 or more glutamines in a row on the Huntington protein after transcription and translation take place. The full reasons of how glutamine affects the body is unknown. However, scientists do suspect that glutamine congregates in the neuronal cells, specifically the putamen and caudate of the basal ganglia regions of the brain, which you can see here and here. It's believed that neuronal cell death is associated with excitotoxicity, which is excessive firing of the neuronal cell leading to high calcium concentrations. The methods for testing this disease just involve counting the number of CAG repeats in the gene. When you have the 10 to 35 CAG repeats, the repeats may lengthen over generations, with the highest likelihood of developing HD associated with repeats of roughly 27 to 35 CAG repeats in the Huntington gene. Symptoms of HD affect the CNS mostly. This includes movement, cognitive function, and drastic mood swings. If enough of the neurons in the caudate and putamen die, then loss of brain tissue volume will increase. This increases the lateral ventricles inside of the brain, with this area being associated with movement, ultimately causing involuntary movement. HD can also cause dementia and depression, all because of the loss in tissue volume in the region of this brain. Finally, due to the death of these neurons, decreased levels of GABA and ACH are also associated with HD, with increased levels of dopamine around this region, leading to few available treatments, which are dopamine suppressants usually. However, they only suppress the symptoms and do not actually affect the survival rate for the patient, only the quality of life associated with the disease, with death commonly associated with aspiration pneumonia due to the loss of coordinated swallowing. There were a total of 79 patients in this study, with an additional 30 healthy subjects used as a control between January 2008 and June of 2016. All patients had a confirmed CAG repeat of 39 or more. All patients underwent an MRI scan of the brain, and the volumes of the thalamus, hippocampus, accumbens, caudate, nucleus, and putamen were taken. Healthy and diagnosed brains were then compared. As you can see, the patients with HD on this slide here are on the left, and the patients who are healthy are right here on the right. If you look at the p-value skipping down to the subcortical structures, you can see that there was a significant difference between most of the previously mentioned areas, excluding the amygdala, meaning that patients with HD have a significantly smaller volume in these mentioned areas of the brain. Like it was said before, the volumes in the accumbens, caudate, putamen, pallidum, thalamus, and hippocampus were significantly lower in patients with HD. Since the mean volumes of the amygdala did not differ between HD patients and the control group, the structures were not investigated any further. Also, TMS and chloria were closely associated, as you can see in this table right here. The UHDRS TMS, which is this top one right here, this top row, was developed as a clinical rating scale to assess 4 domains of clinical performance and capability in patients with HD, which include motor function, cognitive function, behavioral abnormalities, and functional capacity. Here you can see an MRI scan of the brain taken from patients in the study. You can see chloria most resembles the UHDRS TMS in the accumbens nucleus, caudate nucleus, putamen, and pallidum in HD patients. Also, for comparison, I left in patients with signs of dystonia and hypokinesia. The study shown to you today shows how specific clinical motor signs in patients with HD are related to volume loss in different gray matter brain regions. Higher UHDRS chloria scores specifically, recalling that the areas affected were the accumbens nucleus, caudate nucleus, putamen, and pallidum, as to the other regions where it is not as prevalent, meaning that volume loss is more associated with signs of chloria and not cortical atrophy, as previously thought. There is also a known interplay between the basal ganglia and cerebral cortex, especially changes of the basal ganglia thalamo-frontal circuits are known to contribute to hyperkinetic movements such as chloria. In future studies, they should include the relationship between cognitive and psychiatric symptoms of HD. Also, to what extent do the symptoms progress as the CAG extends, say from 38 to 40? Or the speed at which anticipation occurs. Possible errors include the cohort used in the study, which are the 79 patients, such that the patients with HD were earlier into their diagnosis and could not yet exhibit such symptoms. Another experiment in the future could be comparing different age groups to one another, as a means to see how the progression of the disease decreases volume in motor areas and leads to further symptoms.